We have already discussed about the skeletal and the cardiac muscles. Today, we are going to discuss about the smooth muscles. Smooth muscle is distinguished anatomically from skeletal and cardiac muscle because it lacks visible cross striations. Actin, myosin are present and they slide on each other to produce contractile responses. But they are not arranged in regular arrays as seen in skeletal and cardiac muscle. And the striations are as a result absent. Instead of Z lines, there are dense bodies in the cytoplasm and attached to the cell membrane. And these are bound by alpha actinin to actin filaments. Smooth muscles also contain tropomyosin, but troponin appears to be absent in the smooth muscles. The isoform of actin and myosin differ as that of the skeletal muscle. Sarcoplasmic reticulum is present, but it is less extensive than seen in the skeletal or cardiac muscle. Smooth muscles contain few mitochondria and depend to a large extent on glycolysis for their metabolic needs. So we will learn about functional anatomy and organization of smooth muscles, process of excitation contraction, and characteristics of excitation and contraction, and about the excitation and inhibition of smooth muscles. The smooth muscles are non-striated, involuntary, and they are distributed along different organs in form of sheath bundles, say in the wall of the esophagus, stomach, intestine in the GIT, ducts of digestive gland in the trachea. So in different places, smooth muscles are distributed. In the cardiovascular system, you can see them along the blood vessels. In the respiratory system, in the digestive system, in the renal system, in the reproductive system, the smooth muscles are present and their function varies from one organ to the other. So these are involuntary, non-striated muscles. They can be of two types. One is single unit, another is multi-unit. The single unit smooth muscle, the muscle cells are electrically coupled such that electrical stimulation of one cell is followed by stimulation of adjacent smooth muscle cells. This results in a wave of contraction as in peristalsis. In multi-unit smooth muscles, these cells are not electrically coupled. So, stimulation of one cell does not necessarily result in excitation of the adjacent smooth muscle cells. Example, these are present in fast difference of male genital tract in iris of the eye. So, multi-unit and single unit smooth muscle. In the single unit, there are gap junctions present. So, they function as syncytium because they are connected via the low resistance bridges with one another. Now, you can see in this picture, it is the muscular arteriole with fusiform smooth muscle cells in circular orientation. B is superimposed images of circular and longitudinal layers of intestinal smooth muscle. C is rectangular smooth muscle cells with thin projections to adjacent cells in small testicular duct. So you can see different types of different smooth muscles present in different organs. Now look at this picture. We will see about phasic and tonic. Some contractile activity patterns exhibited by the smooth muscles are shown in this diagram. Tonic smooth muscles are normally contracted and generate a variable steady state force. Examples are sphincters, blood vessels, airways. Phasic smooth muscles commonly exhibit rhythmic contraction. Example, peristalsis in the GI tract, and may contract intermittently during physiological activities under voluntary control. Example, voiding of urine, swallowing. In some organs, the smooth muscle cells contract rhythmically or intermittently, whereas in other organs, the smooth muscle cells are continuously active and maintain a level of tone. Smooth muscle exhibiting rhythmic or intermittent activity is termed as phasic smooth muscle. Phasic smooth muscle corresponds to single unit category that we have already learned about. Smooth muscle that is continuously active, on the other hand, is termed as tonic smooth muscle, vascular smooth muscle, respiratory smooth muscle, and some sphincter are continuously active. The continuous partial activation of tonic smooth muscle is not associated with action potential, although it is proportional to the membrane potential. Tonic smooth muscle would thus correspond to the multi-unit smooth muscle that we have already discussed about. Phasic and tonic contractions of smooth muscle result from interactions of actin and myosin filaments. There is a change in cross-bridge cycling kinetics during tonic contraction, such that the smooth muscle can maintain force at low energy cost. They can maintain force at low energy cost. Now, electrical and mechanical activity of the smooth muscle. We will discuss about that. Here you can see in this picture the gap junctions, the myofilaments, the dense bodies which are present in the smooth muscles. Let's see something about the caveoli. You can see here there is a structure that is named as caveoli. So what is that? Caveoli represent invagination of the smooth muscle membrane, analogous to T-tubules in striated muscle. The sarcoplasmic reticulum extends throughout the smooth muscle cells. There are junctional regions of the sarcoplasmic reticulum where it abates regions of the sarcolemma or caveoli or both. These sarcolemmal regions of the sarcoplasmic reticulum play an important role in the regulation of intracellular calcium and hence smooth muscle tone. Again, in this picture, you can see dense bodies, gap junctions, thick and the thin filament, that is the actin and the myosin filaments. Now we will discuss about this picture. Here you can see relationships between membrane potential and generation of force in different types of smooth muscle. Look at the figure A. Action potentials may be generated and lead to to a twitch and larger some mechanical responses. So you can see the electrical response and the mechanical response. Action potential are characteristics of single unit smooth muscles. Gap junctions permit the spread of action potential throughout the tissue. Now come to figure B. Rhythmic activities produced by slow waves that trigger action potential. The contractions are usually reflected the activity. You can see in this figure B, what is happening? There is rhythmic activity produced by slow waves that trigger action potential. The contractions are generally associated with bursts of action potential. Slow oscillations in the membrane potential usually reflect the activity of electrogenic pumps in the cell membrane. Now come to figure C. You can see in this figure, tonic contractile activity may be related to the value of the membrane potential in absence of action potentials. Graded changes in EM are common in multi-unit smooth muscle, example vascular smooth muscles, where action potentials are not generated and propagated from cell to cell. Now come to figure D, pharmacomechanical coupling. Changes in force produced by addition or removal of drugs or hormone that have no significant effect on membrane potential. You can see there is no change in the membrane potential. Still, there is change in force of contraction that is produced by some drug or hormone. So you can compare this four picture. There are different types of contraction and electrical events that are occurring in the smooth muscle. So in the smooth muscle, you know that we do not see a typical neuromuscular junction. What happens when the nerve terminal, it comes near the muscle end plate, the gap between them decreases and there is transmission of impulses which occurs via low resistance bridges, and in the nerve terminals there are formations of dilations which are known as varicosities and they contain the neurotransmitters. So there may be the smooth muscles, they may be innervated by both the autonomic nervous system branches, that is the sympathetic and the parasympathetic. So there may be production of an excitatory postsynaptic potential or inhibitory postsynaptic potential. Now we will discuss about the electrical and mechanical activities of the smooth muscle. Unitary smooth muscle is characterized by the instability of its membrane potential and by the fact that it shows continuous irregular contractions that are independent of its nerve supply. So this doesn't depend on its nerve supply. The membrane potential has no true resting value. And it varies between -20 to -65 millivolt. Smooth muscle cells can display divergent electrical activity. There are slow sine wave-like fluctuations, a few milliseconds or millivolts in multitude, and spikes that sometimes overshoot the zero potential line. And sometimes do not. In many tissues, the spike have a duration of about 50 milliseconds, whereas in some tissues, the action potential have a prolonged plateau during repolarization, like action potentials you have seen in the cardiac muscle. As in other muscle types, there are significant contributions of sodium, potassium, and calcium channels, and the sodium, potassium ATPase pump. Because of the continuous activity, it is difficult to study the relation between electrical and mechanical events in unitary smooth muscle. The excitation contraction coupling in unitary smooth muscle can occur with as much as a 500 millisecond delay. It is a very slow process compared with that in the skeletal and cardiac muscle, in which the time from initial depolarization to initiation of contraction is less than 10 milliseconds. Here it can be 500 milliseconds, this delay can be. Unlike unitary smooth muscle, multi-unit smooth muscle is non-syncytial and contractions do not spread widely throughout. So the contractions of multi-unit smooth muscles are more discrete, fine, and localized than those of the unitary smooth muscles. So you can see here a typical spike potential can be elicited by an external stimulus. You can see this is observed in the GIT, and this is the depolarization is due to mainly influx of calcium. Spike potential with slow waves. Some smooth muscles are self-excitatory. No extrinsic stimulus is required. You can see the basic slow waves of membrane potential. These are not action potential. Now, spike potentials in this picture with slow waves. You can see these in GIT, smooth muscles of intestinal wall. Duration of spike potential is 10 to 50 milliseconds. So, membrane potential of smooth muscle. You can see in this picture, slow waves, there are leaky cation channels. In this picture, you can see there is opening of voltage-gated calcium channel in step 1. This is the causes the spike of the action potential. Now you can see there is influx of calcium. So calcium influx causes rise in intracellular calcium concentration. This causes opening of calcium dependent potassium channel. That is the figure 4. You see in figure 1 there was opening of voltage gated calcium channels, and there was figure 2. You can see there was rise in the membrane potential, which resulted in the action potential spike. Now, as calcium has entered into the cell, what happens? Calcium influx causes internal calcium to increase. The concentration of calcium inside the cell has now increased. In figure 4, what happens? This causes opening this intracellular calcium, increase in intracellular calcium causes opening of a group of potassium channel, which are calcium dependent potassium channels. And this causes slow hyperpolarization of the membrane. You see the figure 5, slow hyperpolarization of the membrane. Now in figure 6, you can see voltage gated calcium channels close and internal calcium gradually decreases. So again, the membrane potential moves more towards hyperpolarization. Figure 7, you can see calcium dependent potassium channels now close. Again, what happens? Again, the voltage gated calcium channels, they start opening. So this is the generation of slow waves. This is the picture of generation of slow waves. So this is the picture of an action potential with a plateau, which we have already discussed about. Now we will see about the mechanism of the excitation, contraction, coupling. It can be brought out by some neurotransmitter. It can be brought out by hormones, a stretch. So there may be different stimuli which can result in change of the sometimes membrane voltage, which results in contraction, sometimes even without a change in the membrane voltage, as we have seen in pharmacomechanical coupling. That is due to effect of some drug or hormone, there may be contraction of the muscle. Now, let us discuss in brief about the molecular basis of the contraction. In the skeletal and the cardiac muscle, calcium, we have seen, plays a prominent role in initiation of the contraction. Now, here also we will see the effect of calcium. Depending on the activating stimulus, calcium increase can be due to influx through voltage or ligand-gated plasma membrane channels. Efflux from intracellular stores through RYR and efflux from intracellular stores through inositol triphosphate receptor IP3R calcium channels, or a combination of these. So the sources of calcium can be from different places. Here in this muscle, there is lacking of troponin, while in the skeletal and the cardiac muscle, we saw there was presence of troponin. Myosin in the smooth muscle must be phosphorylated for activation of myosin ATPase. In the cardiac and the skeletal muscle, we have seen that the myosin head has got auto ATPase activity. It doesn't need to be activated. But what happens? The troponin tropomyosin complex, they keep the active site of the actin filament covered. So the myosin head cannot get attached with the active sites of the actin filament. So the contraction in the cardiac and the skeletal muscle is said to be thin filament regulated. Here we will see in the skeletal, in the smooth muscle, the contraction is thick filament regulated. Here, the active sites of the actin filament, the troponin is absent. So this is not covered. We need the myosin head to be activated for having the ATPase activity. This myosin head has to be phosphorylated for activation of myosin ATPase. Phosphorylation and dephosphorylation of myosin also occurs in skeletal muscle, but phosphorylation is not necessary for activation of ATPase in the skeletal and the cardiac muscles. So this is the basic difference. In the smooth muscle, calcium binds to calmodulin, and the resulting complex is called now the calmodulin dependent myosin light chain kinase. So there is formation of calmodulin dependent myosin light chain kinase, which is getting now activated. This enzyme catalyzes the phosphorylation of the myosin light chain on serine at position 19, increasing the ATPase activity. Myosin is dephosphorylated by myosin light chain phosphatase in the cell. But dephosphorylation may not lead to relaxation because various mechanisms are involved. One is the latch bridge mechanism, by which myosin cross bridge remain attached to the actin for some time after the cytoplasmic calcium concentration falls. This produces sustained contraction with little expenditure of energy, which is especially important in vascular smooth muscle. Relaxation of the muscle presumably occurs when the calcium calmodulin complex finally dissociates, or when some other mechanisms come into play. So what happens? In the smooth muscles? So binding. Let's describe the sequences. Binding of acetylcholine to muscarinic receptors. Same. So increased influx of calcium into the cell. Activation of calmodulin dependent myosin light chain kinase. Phosphorylation of myosin. Increased myosin ATPase activity and binding of myosin to actin. Contraction. Dephosphorylation of myosin by myosin light chain phosphatase. Relaxation or sustained contraction due to latch bridge and other mechanisms. So you can see in this picture the different events that are occurring during the process of contraction in the smooth muscles. Regulation of smooth muscle myosin interaction with actin by calcium stimulated phosphorylation. In the relaxed state, cross bridges are present as a high energy myosin ADP PI complex in the presence of ATP. Attachment to actin depends on phosphorylation of the cross bridge by a calcium calmodulin dependent myosin light chain kinase MLCK. Phosphorylated cross bridges cycle until they are dephosphorylated by myosin phosphatase. Cross-bridge phosphorylation at a specific site on the myosin regulatory light chain requires ATP in addition to that used in each cycle interaction with actin. So the smooth muscle, they can respond to stretch, they can respond to some drugs, they can respond to changes in membrane potential. There may be electromechanical coupling, pharmacomechanical coupling. So these are the steps of contraction in the smooth muscle. You can see here the myosin light chain kinase, calcium binds with calmodulin, and this now activates the myosin light chain kinase, which helps in the contractile process, which will be followed later on. Now, we will also discuss about some properties of the cell and the membrane. Smooth muscle cells lack T-tubules, the invaginations of skeletal muscle sarcolemma that provide electrical links to the sarcoplasmic reticulum. But we have seen there are claviculi in pocketing present here. Claviculi increase the cell surface to volume ratio of the cells and are closely opposed to the underlying sarcoplasmic reticulum. The gap is 50 nanometer. Calcium spark and a variety of calcium handling proteins have been observed near the claviculi. So this can explain the underlying sarcoplasmic reticulum may contribute to the regulation of intracellular calcium in smooth muscle. The voltage gated L-type calcium channel and the 3-sodium 1-calcium antiporter are associated with claviculi. The proteins clavulin and cholesterol are both critical for the formation of claviculi. Calcium can be released from the sarcoplasmic reticulum into the myoplasm when stimulated by neurotransmitters, hormones, or drugs bind to receptors on the sarcolemma. Intracellular calcium channels in the sarcoplasmic reticulum of smooth muscle include the ryanodine receptor, which is similar to that found in the skeletal muscle sarcoplasmic reticulum, and the ionositol 1,4,5-triphosphate gated calcium channel. The RYR is typically activated by a rise in intracellular calcium, that is, calcium-induced release of calcium in response to an influx of calcium through the sarcolemma. The INSIP3 gated calcium channel is activated by INSP3, which is produced when a hormone or hormones bind to various calcium mobilizing receptors on the sarcolemma. Intracellular calcium is lowered through the action of a sarcoplasmic reticulum calcium ATPase pump, that is the SARCA, and extrusion of calcium from the cell by the 3-sodium 1-calcium antiporter and a sarcolemmal calcium ATPase pump. So in these pictures, you can see the contractile mechanisms in the smooth muscle, the latch breach formation, the relaxed phase of the smooth muscles. Now we come to some of the properties of the smooth muscle. Now for relaxation of the smooth muscle, in addition to cellular mechanisms that increase contraction of the smooth muscle, there are cellular mechanisms inside the cell. There are mechanisms which can lead to relaxation. And this is very important in smooth muscle that surround the blood vessels to increase the blood flow. The endothelial cells that line the inside of the blood cells could release a substance that relax the smooth muscles. This is called the endothelial derived relaxing factor, EDRF, which was later identified as nitric oxide. Nitric oxide produced in endothelial cells is free to diffuse into smooth muscle cells for its effects. Once in muscles, nitric oxide directly activates soluble guanylyl cyclase to produce another second messenger molecule, that is CGMP. This molecule can activate CGMP specific protein kinases that can affect ion channels, calcium homeostasis, or phosphatases. And these all together can lead to smooth muscle relaxation. The effects of acetylcholine and norepinephrine on urinary smooth muscles serve to emphasize two types of important properties. One is its spontaneous activity in absence of any stimulus, and its sensitivity to chemical agents released from nerves locally or brought to it by the circulation. Unitary smooth muscles usually have a dual nerve supply from the two divisions of the autonomic nervous system, that is the parasympathetic and the sympathetic nervous system. Stimulation of one division increases smooth muscle activity while the other decreases. In some organs, noradrenergic stimulation increases and cholinergic stimulation decreases smooth muscle activity, while reverse occur in other organs. Latch breach phenomena, we have already told that the smooth muscle can maintain its force of contraction for longer intervals without using much energy due to this latch breach phenomena. So this helps in the conservation of energy. This sustained force is maintained with only 20 to 30% of cross breach phosphorylation, and thus ATP utilization is reduced. The term lax state refers to this condition of tonic contraction during which force is maintained at low energy expenditure. The lax state is thought to reflect dephosphorylation of myosin light chain. When the myosin light chain is phosphorylated, the cross bridges recycle as long as myoplasmic calcium is elevated. However, if an attached cross bridge is dephosphorylated by myosin phosphatase, the rate of cross bridge recycling is decreased because detachment of cross bridge is slower and the myosin light chain must be rephosphorylated before another cycle can begin. When myoplasmic calcium is high, most of the cross bridges will be phosphorylated and shortening velocities of rates of force development will be relatively high. When myoplasmic calcium falls during tonic contractions, the likelihood that a cross bridge will be dephosphorylated and spend more time in an attached force generating conformation increases. A low rate of calcium dependent phosphorylation of myosin light chains is essential for contraction. The muscle will relax if calcium falls below that required for binding to calmodulin and activation of MLCK. Now we will discuss something about the energetics and metabolism. ATP consumption is reduced during the lax state. Under this condition, smooth muscle uses 300-fold less ATP than would be required by skeletal muscle to generate the same force. Smooth muscle, like skeletal muscle, requires ATP for ion transport to maintain the resting membrane potential. All these metabolic needs are readily met by oxidative phosphorylation. Fatigue of smooth muscle does not occur usually unless the cell is deprived of oxygen. Aerobic glycolysis with lactic acid production normally supports membrane ion pump even when oxygen is plentiful. Now we will discuss about the regulation of myoplasmic calcium concentration. We have already discussed about the energetics and the metabolism. So how the regulation of myoplasmic calcium occurs, we will discuss about it. The mechanism that couple activation to contraction in smooth muscle involves two sources of calcium, one involving the sarcolemma and the other involving the sarcoplasmic reticulum. The sarcolemma regulates calcium influx and efflux from extracellular pool. The sarcoplasmic reticulum membranes determine calcium movement between myoplasm and the sarcoplasmic reticulum pool. Skeletal muscle contraction does not require extracellular calcium. In contrast, extracellular calcium is important for smooth muscle contraction. This regulation of myoplasmic calcium involves not only the sarcoplasmic reticulum, but also the sarcolemma. A number of factors can alter myoplasmic calcium of smooth muscle. This differs from skeletal muscle, in which the action potential induced release of calcium from the sarcoplasmic reticulum fully activates and there is contraction that results. Now we will learn about the plasticity of the smooth muscles. Now, force generation and plasticity of the smooth muscles. Now, smooth muscle displays an unique economy when compared to skeletal muscle. That is, we have discussed, despite approximately 20% of the myosin contain, 100-fold difference in ATP use when compared with skeletal muscle that can generate similar force per cross-sectional area is observed in smooth muscle. There are several reasons, that is, unique isoforms of myosin and contractile related protein expressed in the smooth muscle and their distinct regulations. We have already discussed some of them. The unique architecture of the smooth muscle and its coordinated unit also likely contribute to these changes. Another special characteristic of smooth muscle is the variability of the tension it exerts at a given length. If a unitary smooth muscle is stretched, it first exerts increased tension, but if the muscle is held at greater length after stretching, the tension gradually decreases. In the skeletal muscle, cardiac muscle, what you have seen? When the muscle is stretched, the tension increases. Here you are saying initially you are increasing the length, the tension increases. Then what happens? You hold the increased length for some time, the tension gradually decreases. Sometimes the tension falls to or below the level exerted before the muscle was stretched. So it is very difficult to correlate the length tension relationship, and no resting length can be assigned to the muscle. The smooth muscle behaves more like a viscous mass than a rigid structured tissue. And this property that is referred to as the plasticity of the smooth muscle. We will see this with the example of the bladder. The tension exerted by the smooth muscles of the bladder can be measured at different degrees of distension as fluid is infused into the bladder via a catheter. Initially, tension increases relatively little as volume is increased because of the property of plasticity of the bladder. You can see initially there is small increase in the tension. This is due to the property of plasticity of the bladder. So what are the advantages? That is accommodating more blood fluid by the vessels viscera without increasing the pressure, accommodating more urine, say in the bladder, accommodating more food in the stomach. So there are different characteristics. Till now we have discussed about these. One is difference in the structure, unstable resting membrane potential, slow excitation contraction coupling, marked shortening during contraction, latch phenomena, plasticity, less energy required to sustain contraction, can respond to stretch in absence of extrinsic innervation. We have seen this characteristics of the smooth muscle. So the differences we have discussed about the smooth muscle. Now we will see about some of the peculiarities. We will just revise them. Smooth muscle cells are linked by variety of junctions that serve both as mechanical and electrical syncytium. These linkages are essential in cells that must contract uniformly. So the smooth muscles, they need to contract uniformly. The sarcolemma plays an important role in calcium exchange between the extracellular fluid and the myoplasm. The sarcolemma of smooth muscle contains numerous caveoli that contribute to the regulation of the intracellular calcium. The sarcoplasmic reticulum contains an intracellular calcium pool that can be mobilized to increase the myoplasmic calcium. Transporters in the sarcolemmal membrane that regulate myoplasmic calcium include receptor-mediated calcium channel, voltage-gated calcium channel, calcium ATPase pump, 3-sodium, 1-calcium antiporter. Neurotransmitters or hormones can act via receptors in the sarcolemma, can activate PLC followed by generation of second messenger INSP3. INSP3 then activates INSP3 gated calcium channels on the sarcoplasmic reticulum. Many agonists that activate PLC through G protein coupled receptors also activate ROK signaling cascade, thereby increasing the sensitivity of smooth muscle contraction to calcium. There is change in sensitivity of smooth muscle contraction to calcium. Smooth muscle sarcoplasmic reticulum also contain calcium-gated calcium channels, RYR, and calcium reaccumulates in the sarcoplasmic reticulum via the Sarca pump. So we have seen the excitation, contraction, coupling of the smooth muscle, the different pumps, the different hormones, how they act in the smooth muscle for generation of a contraction. Smooth muscles contain contractile units that consist of small groups of thick myosin filaments that integrate with large number of thin filaments attached to the dense bodies. So there are no striations which are present here. Contraction of smooth muscle is dependent on release of calcium from the sarcoplasmic reticulum as well as entry of calcium from outside the cell. Smooth muscle lacks troponin. Phosphorylation of cross bridges by calcium-dependent myosin light chain kinase is necessary for attachment to the thin filament. Dephosphorylation of an attached cross bridge by myosin phosphatase slows its cycling rates. Higher myoplasmic calcium increases the ratio of MLCK to myosin phosphatase activity, which results the more of cross bridges remaining phosphorylated throughout the cycle. This increases shortening of the velocities. Smooth muscle activity is controlled by the autonomic nervous system. It may be controlled by some drugs, some hormones also. A variety of hormone agonists increase the sensitivity of smooth muscle contraction to calcium by reducing the activity of myosin phosphatase. Activation of Rho kinase signaling cascade activity contributes to this inhibition of myosin phosphatase and hence to the increase in the sensitivity of smooth muscle contraction to calcium. So we have seen these. Now in this figure, you can see about the force and the velocity, the relationship, the phasic contraction and the tonic contraction. You can see about them. Now, the response to sustained or tonic stimulation is rapid contraction followed by sustained maintenance of force with reduced cross-beat cycling rates and ATP consumption. This behavior is called as the lax state, is advantageous for muscles that need to withstand continuous external force such as blood vessels, which must be able to withstand the blood pressure. During the lax state, ATP is consumed at less than 1/300 the rate needed to maintain the same force in the skeletal muscle. So the energy requirement is much less. The length tension relationship, hyperbolic velocity load relationship, power output curves, and ability to resist imposed loads are comparable to those of the skeletal muscle. Shortening velocities and ATP consumption rates are very low. Smooth muscles has the ability to adjust the length tension relationship when chronically stretched or shortened. This process is called the length adaptation. Length adaptation. Smooth muscle also have the unusual ability to alter velocity stress relationships, which reflect regulation of both the number of active cross bridge and their average cycling rates for a given load. Smooth muscle is also a synthetic and secretory cell with a major role in formation of the extensive extracellular matrix that surrounds and links the cells. Cellular hypertrophy occurs in response to physiological needs, and smooth muscles can retain the potential to drive. So here in this picture, you can see the process of length adaptation. So skeletal muscle, smooth muscle exhibit a bell-shaped length tension relationship, although the length tension relationship of smooth muscle can vary. Within a short period after stretching the smooth muscle, there is a rightward shift in the length tension relationship such that the maximal force generation occurs at a longer muscle length. Likewise, when a brief time after shortening of a smooth muscle, there is leftward shift of the length tension relationship. The mechanism or mechanisms underlying this length adaptation are hypothesized to reflect a change in number of contractile units in series. So this is about the length adaptation of the smooth muscle. So you have learned about the smooth muscle, the various factors which are responsible for the contraction of the smooth muscle. We will discuss more vividly about smooth muscle in the next class. Thank you.